think we were kind of talking about the membrane, so just remember in regards to the cell membrane that it consists in almost all cells of the phospholipids in which some proteins are embedded, and that makes the inside, that part of the membrane hydrophobic, and the surfaces are hydrophilic. Compounds can cross the membrane either by passive or active transport. We also cause passive transport diffusion, in which chemicals move along the concentration gradient from their high to their low concentration. In case of simple diffusion, the chemicals are nonpolar and hydrophobic, and they can cross the membrane directly. While in case of facilitated diffusion, these chemicals are hydrophilic, polar molecules, and this is why they require either channels or carriers. Now, if you want the diffusion, the movement down the concentration gradient does not require energy. Active transport, though, does. In case of active transport, molecules are transported, for the most part, against the concentration gradient. Usually from their low to their high concentration. Okay? Active transport always requires energy and always requires transmembrane proteins. Now, we are getting into the parts of the cells that are optional. Inclusions, for example, shown here, are basically accumulation of chemicals within prokaryotic cells that serve different purposes, from nutrition to orientation in space. Plasmids, which pictures I don't have here, are small, circular, double-stranded DNA molecules that can carry beneficial genes. For example, genes for antibiotic resistance. Plasmids are not essential for the cell survival. And then we have endospores. Endospores are a very sturdy structure shown here that are formed under harsh environmental conditions. During sporulation, the formation of endospore, genetic material of a cell is protected by multiple layers from any pretty much environmental challenge. Okay? And endospores can survive not just for years, not for centuries, but for thousands and millions of years. When endospore ends up in the environment that is good, food, water, endospore germinates, forming a vegetative cell. So sporulation is the formation of endospore, germination is the emergence of vegetative cell from an endospore. This is clinically relevant because of a lot of human pathogens. They, form endospores. Okay? Sounds good? Most bacteria have cell walls. So cell wall is located outside of the membrane, as you can see here and here, and it consists of long carbohydrate chains, so-called glycan chains. So glycan chains are represented here. And these glycan chains are connected by peptide bridges. Remember, peptides consist of amino acids. This is why we call this structure peptidoglycan. Peptido and glycan. So this peptidoglycan cell wall is different in different types of microorganisms. This is why we call some bacteria gram-positive and some bacteria gram-negative. Gram-positive bacteria have a thick layer of peptidoglycan just outside of the membrane. So their cellular envelope effectively consists of two layers, a membrane and a wall. Gram-negative microorganisms have a fairly thin cell wall right outside the membrane, and then outside of that cell wall there is another so-called outer membrane, which contains several important molecules such as lipopolysaccharides, molecules that can cause disease in humans, porins, molecules that facilitate transport. Now, the wall of gram-positive microorganisms also contains important molecules such as teichoic acid, which contributes to the adhesion of these cells to the surface. Overall, if you will compare gram-positive and gram-negative cell membranes, you can see the difference. Gram-positive has 2 layers and gram-negative has 3. Gram-positive contains teichoic acid, while gram-negative membranes contain lipopolysaccharides. Okay? Our attention to cell wall is not accidental, because a lot of antibiotics, such as penicillins, cephalosporins, bacitracin, vancomycin, they target the synthesis of the cell wall. Now, especially in gram-negative bacteria, this molecule called polysaccharide, lipopolysaccharide here, which consists of several sugar chains, carbohydrate chains, and fatty acid molecules, can trigger a very powerful immune response, very strong inflammation, okay, causing non-specific generalized symptoms. Not all bacteria have cell walls. A group of molecules, such as mycoplasma, lack the cell wall entirely. And not all bacteria have either gram-positive or gram-negative cell walls. If you will compare the cell wall of bacteria such as Mycobacterium tuberculosis, shown here, they do look like gram-positives with a thick peptidoglycan cell wall, but on the outside they have a layer of a pretty waxy compound called mycolic acid. It's effectively wax. Okay? One of the common things to mix up: bacteria that contain mycolic acid, mycobacteria, and mycoplasma, cells that do not have cell wall at all. Outside of the cell wall, some bacteria have a layer of carbohydrates called glycocalyx. You need to know 2 types. One type is capsule. It's a pretty sturdy layer that facilitates the adhesion of bacteria to the surfaces, making them more dangerous, and also protects bacteria from phagocytosis by your own immune cells. A more loose version of glycocalyx is a slime layer. It doesn't protect bacteria from phagocytes, but it does increase the adhesion, facilitating the formation of biofilms on the surfaces. Biofilms, most bacteria exist in the form of biofilms, such as bacteria on the surface of your teeth, walls of your gut, as well as bacterial biofilms can be formed on the ventilation tubes and catheters in the hospital, presenting a very major hospital-associated infection threat. Many bacteria can move, not all of them. They can do it using a flagellum. You don't need to know the exact details of the flagellar structure between, in a gram-positive versus gram-negative microorganism. You need to understand this, and it's very simple. The way flagellum operates is basically you have a rod with a hook, and at the end you have the filament. And when rod turns, the filament works as a propeller. That makes sense? That's it. Bacteria can have one flagellum. In this case, we call them monotrichous. 2 flagella on the opposite ends of the cell. We call them amphitrichous. Lophotrichous bacteria have multiple flagella, one side and peri. Trichous bacteria have flagella all over the surface. In addition to flagella, another external appendage, which unfortunately or fortunately does not facilitate the movement but increases the adhesion to the surface, is fimbriae, a bunch of hair-like structures on the surface that is sticky. And the third appendage that I wanted to mention is pili. Pili are long, tubular structures that can either facilitate adhesion or enable bacteria to exchange DNA, so-called horizontal gene transfer. These are called sex pili, and they are important because can increase the spread of antibiotic resistance. Now, go ahead.
Are the fimbriae and the pili, are those types of flagella?
No, that's a good question. No. Those are three absolutely independent, structurally different appendages. Flagella move, fimbriae adhere, pili adhere or exchange genes. Now, when bacteria move using a flagellum, then can either run or tumble. When they run, they move into a certain direction. When they tumble, they orient themselves according to the stimuli. What can be the stimulus to movement? It can be chemical, light, or even magnetic field. Now, what do we call movement? We call movement taxis. So if a movement is driven by chemicals, we call it a chemotaxis, right? Light, phototaxis. Bacteria can move towards something or away. If bacteria move towards a chemical, like a nutrient, we call it positive chemotaxis. If bacteria try to avoid light, that would be negative phototaxis. Okay, we're good? Now, if you start looking at the immediate source of energy for flagellar movement, you will be probably surprised that it's not directly ATP. It's a concentration gradient of hydrogen ions across the membrane. I'm pretty sure you're asking yourself, why is that relevant? Why is that important? Well, hydrogen ions are effectively acid, effectively pH. If bacteria ends up in the environment with suboptimal pH, it may lose the ability to move because of disturbed concentration gradient. But it's a movement of hydrogen ions, a diffusion of hydrogen ions, very much like in the oxidative phosphorylation, that facilitates the movement. So probably the poster child of bacterial infections is Staphylococcus. So we're going to talk about 3 species. I'm going to knock out Staphylococcus epidermidis, which is a normal microorganism present on human skin. It's not really a pathogen. Staphylococcus saprophyticus is a very narrow area of pathogenicity. It's found in the human gut and transmitted mostly sexually during anal sex. And when it infects, especially men, male reproductive and male urinary systems are kind of merged at some point. So that may result in UTI, urinary tract infection. Men can pass it to women, and then it's urinary infection in them. The most notorious, the most well-known is Staph aureus. So it causes a variety of infections. Here is the illustration of what it can do on the skin. It can start with a fairly mild superficial skin infection known as impetigo, which is contact transmitted, and eventually develop into so-called scalded skin syndrome because bacteria start breaking down the connection between the epidermis and dermis, and skin is just falling off. If it infects hair follicles, it can result in furuncles and carbuncles. This is a furuncle. This is a legitimate carbuncle. Okay? When it starts crawling subcutaneously, it can cause cellulitis, but the most notorious, of course, is this. This is the necrotizing fasciitis, flesh-eating disease. Okay, it is treated by surgical removal or removal using sterile maggots, debridement, basically antibiotics, very few times. As I mentioned, for all microorganisms that we mentioned in the course, there are some fundamental things that you must know just by default. Disease, transmission route, vaccine. So transmission route for Staph aureus, either contact or respiratory for infections like sinusitis, not UTI, obviously, sinusitis. And it also can be transmitted through blood. Toxic shock syndrome is the result of hyperactivation of human immune system by one of the toxins of that microorganism, and it was discovered when a bunch of women got sepsis by using female hygienic products, namely tampons, that contained naturally derived cotton. And this cotton was contaminated with Staph aureus. It entered the bloodstream and they had sepsis and many of them died. You don't need to know virulence factors, don't worry. Okay? Now, eukaryotic cell. I'm not going to go into much details. Let me explain why. It's probably 10th time we talk about the parts of eukaryotic cell. I will 100% ask you to match the part and the function. The part and the function. Okay? Now remember I mentioned the importance of terminology. Right? Terminology is important. Nucleus, nucleolus, and nucleoid are three different things. Just to keep in mind. Okay? So, I'm going to get back to the quiz. Okay, plasma membrane. Similar ideas in bacteria. Okay? Phospholipid bilayer, cholesterol to regulate fluidity, transmembrane proteins as signaling proteins or transport molecules, peripheral proteins, usually interacting with integral proteins as well. Okay, one sec. Perfect. So that's all pretty straightforward. Now, what about the maintenance of the cell shape? What about the cytoskeleton? So cytoskeleton in eukaryotic cells consists of microtubules, for God's sakes, that are made out of tubulin. Okay. Microtubules are dynamic, which means they can be destroyed and rebuilt. They're important for intracellular transport, for movement of cells, and for the cell division. Microfilaments consist of the protein actin. They're also dynamic, meaning they can be rebuilt, can be destroyed, and participate in similar functions such as movement, cell division, and sometimes transport. Unlike these two, intermediate filaments that consist usually of the protein keratin are stable. They're destroyed, means cell collapses. They are like beams and studs in the house. So you need to know what these filaments are made of, right? And whether they're dynamic, stable, and what their function is. That brings us to mitochondria and chloroplasts. Well, first of all, this is indeed the powerhouse of the cell. Mitochondria used to be an independent aerobic bacterium that could perform aerobic respiration. They were acquired billions of years ago by first eukaryotes, and both parties benefited from that relationship. Eukaryotic cells found the source of ATP. Okay? Bacteria received protection and constant supply of nutrients. We doing all right so far? Same story is for chloroplasts. Those were photosynthetic bacteria billions of years ago that got accepted by that first generation of eukaryotes. Okay? Why do we think it is that? Why do we think it was endosymbiosis? Both of them can undergo binary fission. When eukaryotic cells divide, mitochondria and chloroplasts also divide. They're not synthesized from scratch. You can replicate them even in an egg. They have DNA, which is very similar to bacterial DNA, and they have ribosomes, which are effectively bacterial ribosomes. All that evidence, albeit circumstantial, pretty much confirms that they are endosymbionts. Similar to bacterial capsules, some eukaryotic organisms have extracellular matrix, which effectively can form a capsule. For instance, pathogenic fungus, Cryptococcus neoformans, forms a capsule around the cell. Mostly proteoglycans in the protein collagen. Eukaryotes also have flagella. They look the same as bacterial, but structurally they're different. Remember how I compared bacterial flagella to a propeller? This is an oar, a paddle. Instead of moving in a circular fashion, flagella of eukaryotes, they wave. Cilia are structurally similar to flagella, but they much shorter, more numerous, while serving the same purpose, movement, either a microorganism or moving food into, the part of organism that actually eats it. Okay, sounds good? So let's chat about some of the pathogens, I think, which is going to finish week 1 and then pop in the lab and do the lab. It's going to take, it's not a lot of time. So first category of eukaryotes, protozoa. You must know this classification. The classification is based on the movements. Ciliophora move using cilia, which is like, yeah. Amoeba move using pseudopodia. Mastigophora use flagella, while apicomplexa can't move at all. Now, if you look at the third column in this table, only apicomplexa group has no species that live independently. All of them are parasitic. And that is exactly because they can't move. If you can't move, you can't chase food. You have to have food around you. Now, I mentioned before, you don't need to know the examples. I just provide them to you. And in this example, just to show, if the color of the example is blue, it's not parasitic. If it's red, it's parasitic. What about this guy, for example? It means that it's non-parasitic, but can be. And that brings us to one of my favorite organisms, Igleria fowleri. It's an amoeba. These amoebae exist in soil and in stagnant, more or less stagnant waters, like ponds and lakes. When human kids swim in the water, they can snort some of the water, and bacteria, this amoeba can go through the roof of the nasal cavity, through the cribriform plate, and ethmoid bone, straight into the brain, resulting in primary amoebic meningoencephalitis. It's a very quick disease, and I want you to understand, for amoebae, it's the dead end. They trapped. It is not a communicable disease. If somebody has amoebic meningoencephalitis, they do not pass it to anybody else. It is often misdiagnosed as meningitis, treated with antibiotics, which means people lose valuable time. In most cases, result in death. Some cases result in survival because it is treatable if it's early, but it leaves a long-lasting neurological damage. It's not frequent. I don't remember which year I picked up that statistic, but 1962 to 2008. So we're talking 46 years, we have 111 cases, not a lot. Some states don't have these cases at all, which is totally explainable. This is what I love about epidemiology. If you look at the graph, people who get infected the most, kids. When they get infected the most? Summer. Where? South, because it's super hot. And remember, the map shows the total number of cases. So there's no surprise that Texas has the greatest number of cases because there are so many people. And it shouldn't come as a surprise to you also that Florida is the second because, well, it's Florida. Okay? Now we jump to another eukaryote type, fungi. Good news. Now, the only classification that I want you to use, and I know that it's not a proper taxonomy, but it's useful. Difference between molds, yeasts, and dimorphic fungi. Molds are multicellular, although cells confuse sometimes. They form hyphae, that fuzzy appearance, right? While yeast are unicellular. These chains of cells actually are chains of individual independent organisms. Now, all of them can reproduce by fragmentation, hyphal fragmentation or budding in yeast. All of them can reproduce by spores, either sexual or asexual. Now, what about dimorphic fungi? So at the ambient temperature, let's say 73, 74, dimorphic fungi grow as molds. When they get in the human body, though, which is much warmer, right, they start growing as yeast. A lot of pathogenic fungi are dimorphic. One of the examples is here. Histoplasma capsulatum, also known as Ohio Valley fever. It's a fungal infection that is transmitted by inhalation of spores from the environment. The spores usually come from feces of bats or birds. When these feces get aerosolized, people inhale them. As a rule of thumb, people who are immunocompetent rarely have noticeable fungal infections except for the skin or like thrush in the mouth. That makes sense? Anything systemic, no, because they're healthy. But in immunocompromised individuals, this fungus can cause pneumonia, can spread systemically, and if it does, and if it is untreated, it is uniformly lethal. It can be treated. There's a bunch of antifungals. It's not, an inevitable thing. It's not common. As you can see, there's a reason why we call it Ohio Valley fever. It's Midwest. I'm really curious why there's no histoplasma to the west of Rocky Mountain. That is really curious. I don't know. I don't have an answer. By the way, people who are very much at risk are lumberjacks. Right? A lot of bat and bird poo. All those trees fall down, aerosols everywhere, so they are advised to wear PPE. And my second favorite organisms in this course, helminths. They are absolutely adorable. You need to know 3 major taxa, major phyla. Nematodes, trematodes, and cestodes. And you need to understand what's the difference. Nematodes, roundworms with a cylindrically shaped body. They don't have segmentation. They have proper sexes. And they have a pretty developed organ systems with a body cavity. Trematodes, also known as flukes, are flatworms that are not segmented. They are hermaphrodites, and they lack body cavity. Finally, cestodes are the simplest of these worms. They flat. They are segmented, which I'm going to demonstrate in a minute. Also hermaphrodites, and lack body cavity as well. Effectively, these guys, tapeworms, feed themselves by diffusion of nutrients from the gut of the host. And my favorite of all helminths, I'd say, is a pork tapeworm, Tinea solio. So this is what its life cycle looks like. It's actually quite fascinating. We start with either an egg or a fragment of a tapeworm called proglotid in the soil. When an egg or a proglotid, which is full of eggs, is consumed accidentally by a pig, we're not talking about cows now, by a pig, this egg hatches. The larva and hatches in the intestine. The larva penetrates the intestine and ends up in the pig's muscle. Did you ever buy pig's muscle? It's called pork. Okay? So when this pork is consumed then by a human being, especially, well, not if it is undercooked, the larvae will end up in the intestines of a human, develop into an adult worm, which will use these little hooks on its head to attach itself to the wall of the intestine, and then it's just going to leach a bit of your food. Not a lot. You're not going to lose weight. Most likely, you're not even going to notice. Most people with tapeworm infections are absolutely feel fine. Maybe there's some minor abdominal discomfort. Okay? It tries to parasitize on you as little as possible. Okay? Now that doesn't seem too awful. Like, why are we concerned as much? Well, it turns out if somebody has tapeworm, tapeworms produce a lot of eggs. Hundreds of thousands of eggs a day, actually. These eggs then passed in feces. As allegedly one of virologists, Robert Schopp, had a plaque on his door saying that the world is covered with 3 inches of feces, meaning that fecal contamination is everywhere. And he was right. So when these hundreds of thousands of eggs come out with poo, when you flush, some of them aerosolize. They end up everywhere, including your hands and so on and so forth. You carry them out in the field, pork, pigs, they eat it, blah, blah, blah. The cycle continues. But what if a human eats such an egg? Human and pigs are pretty close. So if this egg goes into human gut, it hatches, forms larvae which penetrate the gut and end up in the muscle, but what's more important, they end up in the brain. This condition is called cysticercosis. If it's a muscle, it's myocysticercosis. If it's a brain, is neurocysticercosis. It is one of the leading causes of epilepsy in the world. Okay? One of my favorite cases was in New York when a family of Orthodox Jews caught neurocysticercosis, which, if you're familiar with Jewish dietary traditions, sounds very suspicious. Turns out it wasn't them who violated Torah, Talmud, whatever. They had a housemaid, which was from Mexico. She was great. But it is known that a lot in Mexico, especially in the rural area, there are villages where all people carry tapeworm. So she did. When doctors were taking samples of the environment, they found eggs in tapeworm eggs in the dust on the windowsills. There were so many of them. So it was a question of time. Everybody got treated, everybody got fine, like it's treatable. Okay? Cooked pork. All pork that goes into the United States is checked. Obviously, you can't check each and every pork chop. But it is checked. There's monitoring. I can tell you I kind of worked with USDA when I was in research. They do not mince words. This is one of the most, I don't want to say evil, but they are very picky and very attentive, and they have some laws that can, like, there are some laws that can put a person in jail for felony if they violate certain agricultural norms. Like, it's pretty, this guy's pretty serious. Okay, and the last bit for today, the viruses, my first, most favorite microorganisms. As we mentioned before, they are acellular, which means they are non-living. Okay? I don't know. There we go. Acellular, non-living. If there will be every question on like a board exam, a virus is alive, the answer is no. Good? Most viruses are absolutely benign. There's like under 300 viruses that cause human disease known now. They are obligate intracellular parasites. They can't reproduce outside of the cell. They can exist. You can have viruses like sitting on that table, but they're not going to reproduce. Viruses completely rely on cellular ribosomes. They don't have them. And they rely on cellular metabolism. They don't have their ways to generate ATP. Structurally, generally, viruses are divided into naked viruses, which consist of the nucleic acid genome and protein capsid, and enveloped viruses like this HIV, which have a nucleic acid genome, a capsid, and the envelope that consists of phospholipids stolen from the cell and viral proteins. This information may sound boring, probably, but not for me, but you know. But it's quite important from the standpoint of microbial control. Turns out, if a virus has phospholipid envelope, it's more sensitive to various chemicals such as hand sanitizer, alcohol. Naked viruses without that layer are more resistant. I know it sounds counterintuitive, but we'll get to the explanation why. So, for instance, rhinoviruses that cause common cold are naked viruses. The only thing that can prevent the transmission is hand washing. I mean, obviously, not interacting with people also helps, but hand sanitizer doesn't work. That make sense? They were discovered by a Russian scientist in late 19th century, Dmitry Ivanovsky. He called them initially filtrable agents because they were passing through the filter that was impermeable for bacteria. So he said that they were filtrable infectious agents. First was a plant virus, tobacco, mosaic. They widely range in size, from hepatitis D, which is super small, 20 nanometers, to Pandora virus that can be seen in the light microscope, 700 nanometers. It's pretty huge, okay? So papillomavirus, I chose it not because I don't really like it, like it's not one of my favorite viruses, but it's kind of, you know, hits close to home. So it is a virus. Oh yeah, viruses, since they're not alive, they don't really have a restriction on the type of genome that they have. DNA, sure. RNA, sure. 2 strands, no problem. 1 strand, no problem. So anything goes. So papillomavirus has a double-stranded DNA genome. It's a naked virus, so no envelope. And to my knowledge, it is the most resilient, like resistant to chemicals, among viruses that infect humans. you have to have like pretty concentrated bleach to inactivate this virus. It infects stratified squamous epithelium. I'm not going to torture you asking if you remember. In P1, we're talking skin, stratified squamous, mouth, stratified squamous, reproductive organs, stratified squamous, glans, penis, or vagina, and anus, because it also is covered with a stratified squamous epithelium. When this virus infects that epithelium, it goes into the basal layer. Are you with me so far? The layer in the epithelium that actually divides. Because virus needs cellular division to reproduce. It needs cellular enzymes, stuff like that. So it forces cells to divide. When cells divide, there are more cells. Agreed? If it's a skin, this accumulation of cells, we call it a wart. It's benign, can be removed. Okay? Warts can be on the skin, they can be on the anal epithelium, they can be on the vaginal or penile epithelium. So vaginal warts, penile warts, anal warts, no. Okay? But if you force the cell to divide often and to replicate its DNA, this DNA can accumulate mutations. Are you with me? These mutations may lead the cell on the path towards becoming cancerous, which is not good. Cells have mechanisms to kill themselves, just like Jeffrey Epstein did, when they become close to malignant. Okay? So, for some inexplicable reason, some subtypes of human papillomavirus turn off that safety switch, turn off that pathway to apoptosis, to death. And those cells keep going and eventually become malignant. The most known, cancer is not essential. You have to understand that. It is not like, virus doesn't need cancer for anything. By the time somebody develops cancer, it's spread to somebody else. It's done. It's collateral damage. Cervical cancer. Practically all cervical cancer is caused by HPV infection. It also can cause penile cancer, anal cancer, vaginal cancer. It is implicated in certain cancers of head and neck, basically throat cancer, where it kind of works together with Epstein-Barr virus. My point is, we have a pretty good vaccine called Gardasil. One of the best vaccines humans ever made. It is, so its structure is such that it is ultimately safe. It can cause, like, obviously, pain at the site of injection, sure. There are no severe side effects at all. It is recommended now for kids before they start active sexual life, like age 11, 12. It is recommended for adults, and I think I just passed the age. I think it's until either 40 or 45, I don't remember. After that, it's like, nah, no, makes no sense. Okay? But I just want you, like, you're going to interact with people professionally. You're going to talk to parents. One of the concerns that people expressed about that vaccine, if you vaccinate a teenager, that then they become just having sex everywhere with everybody. There is no hornier thing than a teenager, period. It does not depend on the vaccine. Vaccine changes nothing. If you think vaccine will make them, lack of vaccine will make them not having sex, you're wrong. So that is not a legitimate concern. And it's extremely effective. And it's been shown that the rates of cervical cancer has been plummeting. Australia has a program to eliminate cervical cancer, as well as Sweden, by 2030, sorry. Just now, because the compliance is pretty high. Good? Make sense? So, thank you for being patient with me. I'm going to, over the weekend, try to work out how can I get more space.